Hello, everybody. Thanks for coming. We'll go ahead and get started as people kind of continue to trickle in. I'm very excited today to welcome Dr. Anieke Brombacher, who is a postdoc at Yale, to give our talk. Anieke does really awesome work on foramen evolution, or on evolution in general, but using Foraminifera to understand the mechanisms and processes that drive evolution in the marine realm. She is a postdoc at Yale right now. Before she was at Yale, she was also a postdoc at University of Southampton in the UK. Southampton is where she did her PhD in micropaleontology. And she also has a master's in biogeology from the University of Utrecht and a bachelor's of science in math, also from Utrecht. And so I think all of those different experiences of training are coming out in this cool study using a lot of mathematical techniques to understand the evolution of Foraminifera through time. So I think this is going to be a really cool talk, and I'm excited. And welcome to UT. Yeah.
Thanks, Chris, for that welcome. And also thanks for having me over here. It's very nice to visit. Yeah, so as Chris already mentioned, I'm a micropaleontologist, and I use forams to study evolutionary processes because they've got such an amazing fossil record. Chris has already mentioned some of this, but that's okay. Started out as a mathematician at Utrecht back in the Netherlands. Really loved maths, but after about 3 years, things just get so incredibly abstract, like you don't know where the real world comes in anymore. You start to lose touch with reality a little bit. At the same time, I was doing some extra courses in the Department of Earth Sciences in Utrecht. And when I saw my first foram down a microscope, I was hooked. So I decided to do a master's with them, basically using forams to reconstruct past environmental changes. And then for my PhD, I stuck with forams, but using them for evolutionary applications instead. As for my PhD, mainly looking into extinctions and what was driving extinction, whereas the postdoc was more into speciation processes. And now in my current postdoc at Yale, I'm kind of combining all of that together to look into the the climate drivers of evolutionary change, and using mathematical statistical tools to sort of quantify the links between them. I only started at Yale a couple of months ago, so I'm still very busy collecting data. Don't have a lot of results yet. So most of my talk will be about my postdoc from Southampton, about speciation processes. And at the end of the talk, I'll show a couple of slides of what I'm doing at Yale and like possibilities for future work. So for my postdoc, I looked into the effects of developmental plasticity on speciation. And developmental plasticity is the process where a single genotype can give rise to multiple different phenotypes depending on the environment. Baby sea turtles are a really good example of that. The male to female ratio of hatchling sea turtles depends on the temperature of the nest. So the higher the temperature, the more females you get. That's a pretty extreme example. There's much more subtle examples as well. For example, human height and weight as adults depend partially on the nutrition that we receive as babies and as children. And generally, plasticity is thought to have an influence on evolution, because with all these extra phenotypes, you have more like possibilities for natural selection to act on. But it's very difficult to sort of prove that this actually results in speciation processes. So usually what people do is they look at modern species, and they use like the oldest species as proxies for the ancestor, and then the youngest species as proxies for the descendants. So this is an example of spade-foot toads. So this particular study looked at these white ones. Those are the sort of used as the oldest species, so the proxies for the ancestors. And then in black, we've got the descendant species. The ancestors are like omnivores, very generalist. They have a very sort of simple gut morphology. So this is the gut of the tadpoles. And then the more specialized ones, they only eat shrimps. And their gut is a lot more specialized, much more complicated. And what these authors realized is that these the ancestor species, they seem to be a little bit plastic in how their gut morphology looks depending on what they eat. So if you only feed them shrimp, they start to look a lot more like these guys. So they repeated that for a couple of generations, just fed the generalist species, loads of shrimp, nothing else. And then after a couple of generations, they really started to look like these guys. So they hypothesized that this sort of plasticity could have resulted in a much more specialized species if, for example, the environment was such that they only had shrimp to eat. But the problem with that is that we don't know whether that actually happened, because we don't know what this ancestor here at the bottom, what that was actually like. We don't know if it had this opportunity to like change its gut morphology depending on the food that it got. And we also don't know what the food was like here. So we don't know if there was an environment that these guys ended up in that only had shrimp. So whether plasticity was the result of speciation in this case, we will never be completely certain. You really need like deep time data for that. But then frogs, and especially tadpoles, they don't really fossilize that well. And then we also don't know anything about the environment. So this sort of study tends to be really hard simply because we usually don't get ontogenetic trajectories of fossil the fossil record doesn't usually have a lot of developmental data. And then to be able to tie that to the direct environment that the specimen grew in, we don't usually have that. But planktonic forams are an exception. They've got an amazing fossil record, really high resolution. If you take samples through a sediment core, you can literally see species changing and splitting up into new species. And they have this really handy thing where they preserve all of their developmental stages. So it starts in the middle here, what you just saw. The smallest chambers, those are the first ones that are formed. And then throughout its life, it adds a new chamber every few days. So we got the entire ontogeny, so we can reconstruct what these things look like at all the different times in their life. And at the same time, we can also reconstruct the environment from the shell chemistry. So to record environmental proxies in the shell chemistry. So we can directly link developmental processes to the environment. Nobody's done anything like this before. So the first thing we needed to do to study plasticity in forams is to figure out which traits are actually biologically meaningful. So we started with a tool from modern ecology. They're called integral projection models. And they're basically modeling population growth rates depending on different life history traits. So reproduction, survival, that sort of stuff. This model is really nice in that it assigns different chances of, for example, survival and reproduction to different life stages. So you get a really like good representation of the individuals throughout their life. And then you can figure out which of these traits have the biggest influence on population growth. So we studied 4 traits here. Survival, that's basically just the chances of survival at different life stages. Fertility, that is how many offspring every individual produces. Then we have growth, which are individual growth rates, so how quickly every individual grows. And then recruitment is recruitment to the breeding population. So that is the percentage of offspring that are having offspring of their own. So basically the percentage of offspring that's successful. And then on the y-axis, we have the impact that every of these traits has on population growth. And we found that individual growth rates are by far the most important thing for population dynamics, which was great for us because this is the only thing you can measure on fossils, like getting survival or fertility. Like you're never going to get that from the fossil records. So we were really happy with this result. And then the next thing we could do with the integral projection model is figure out how variation in growth rates influences the population. So variation, seeing like what as a sort of measure for plasticity. If growth rates are very plastic, responding to the environment, what influence does that have on population growth rates? So we've got 2 different measures for variation in individual growth rates on the x-axis and the y-axis. And then these colors represent population growth rates. So the redder the color, the darker the color, the higher the population growth rate is. And here it looks like that the more variation in growth rates there is, the higher the quicker the population grows. So based on just this, we would expect that forams are infinitely variable because that just means that is the best for the population. But we don't really see that in the fossil record. Anyone who just starts out picking forams knows it can be quite hard to even tell different species apart because they're all quite similar. It's all popcorn, lentils. So there must be something that is holding them back. It could be that there's ontogenetic constraints, just like architectural constraints in the shell. People have looked into that before. Just the general how these things are built has been fairly constant since the Jurassic. So it could be that there's internal constraints that are holding these things back as well. So for my project, I wanted to look into how we could tell apart plasticity and sort of like genetic variation and internal constraints in species, and how they both contribute to the speciation process. So we studied this lineage of Pliocene Menardella species. They look fairly similar just from these pictures, but there's some key differences between them in terms of how high the shell is, how many chambers they have. So yeah, they're all closely related. And then we took a bunch of samples. So these dotted lines here are the samples that we took through the speciation events, just to see where the differences arise between the species and whether they might have been driven by the environment or by internal constraints. So we started just with measuring individual growth rates. Then we looked at the three-dimensional growth trajectories as measures for shell shape. And then finally, we reconstructed the environment from all the individual shells just to see what, for example, sea surface temperature was like at the time that they were growing, and if that had an effect on growth rates. So to get to the growth rates and the developmental trajectories, we used micro CT scanning. This was our very high-tech setup. So this tube is a see-through cocktail drinking straw. We ordered a box of about 1,000 of these to the apartment, and we had to explain to reception that we were not hosting an illegal party. And then these green and gray bands, they are flower arranging foam. So, you know, the stuff that you stick flowers into, keep them up for a while. We chose that stuff because it's very light. It's got a completely different density from the forams. So when you're CT scanning, you can tell apart what is a foram because it's very dense, and then everything else you can just discard. And then we use different colors to just show where our forams are so that we knew where every individual was. And then after the scanning, we took them out and we used them for chemical analyses for the environmental reconstructions. So we had about 55 specimens in total, and all of these had to be analyzed manually to reconstruct all the individual chambers. So this is a cross section of one specimen. So all of these colors represent individual chambers, and then every foram. Every CT scan of a foram contains about 300 of these slices. So you can imagine this is an incredibly time-consuming process to reconstruct all the chambers of all these forams. Our research technician, Alex Barnes, took the lead in this, and he had help from a couple of master students who basically spent most of their lockdown just marking up foram chambers. So yeah, we got quite a good number of specimens out of that. So when we have all these chambers reconstructed, we know what their sizes are. So here on the left is just back. There. So on the left is just the chamber size of the individual. So you see it increases, but it's a little bit wonky. Also, one thing to note is that the first chamber is always bigger than the second chamber and nearly always bigger than the third chamber. So that was quite interesting. And then we use that data to reconstruct the cumulative volume, which is the total shell volume at every ontogenetic stage. So for example, at chamber 10, the cumulative volume is just the first 10 chambers combined. So you see what the whole foram had to live with. And you can use that to reconstruct growth rates. So that is just the increase in total shell size from one step to the next. And you can see it's quite high in this sort of bit of the ontogeny, and then it sort of slows down a little bit towards the end. So we use that as a proxy for growth rates. Next thing, the shape measures, we had to reconstruct three-dimensional growth trajectories. We used, again, the CT scans for that. So when you've got all these individual chambers reconstructed, you've got their size and their shape, but you've also got things like centroid coordinates, so the coordinates of the center of each chamber. And that's what's plotted here. So all of these numbers are individual chambers. That little dot in the middle is the centroid coordinate. And then you can reconstruct these growth spirals, which are really fun. You can do lots of sort of three-dimensional geometry with them. To speed up this process a little bit, I wrote an R package that can automatically analyze all of these growth spirals. It just needs the centroid coordinates, and then it calculates things like angles between chambers, distances between chambers. It can put chambers in the correct order. It can calculate just how high the spire is. So there's quite a lot you can do with it. Anyone who's interested in this sort of stuff, the package is openly available on my GitHub page, and I'd be happy to talk through this if anyone is interested. So yeah, just a couple of things you can do with it. Chamber angles, like angles between consecutive chambers. You can count how many chambers there are in each whirl. You can calculate trochospirality, which is sort of how quickly the shell goes up into the vertical or whether it's really flat. Trochospirality in particular is quite interesting because it's something that's quite used in atlases as a sort of qualitative measure. But for as far as I know, no one's ever tried to quantify it. So this could be a way to help species taxonomy. But for the rest of this talk, I'll just focus on the number of chambers in the final whirl and sort of the angles between the chambers, because they turned out to be the most descriptive for these particular species. So these red dots are all the individual chambers, and then the final whirl is pretty much all the ones around the outside. Again, it's used as a taxonomic descriptor quite a lot in atlases to tell species apart. So I thought I'll just start with that and see where that gets us. And for this project, I wanted to know if this number is constant through ontogeny. If it is, that means that species are growing isometrically, so they look the same throughout their life. If not, that means there is something that they adjust throughout their life to deal with whatever is going on in their environment. So that's what we started with. And as we found out, these things do not grow isometrically. So these are the chamber centroids again. So here, final chamber, chamber 19, has 7 chambers in the final whirl. But then earlier in its life, at chamber 16, there were only 6 chambers in the final whirl. And even further back, there were 5. So forams don't grow isometrically. Something happens that makes them slow down their growth. It could be that there is a constraint on how big they can actually make their chambers. If they were to keep growing exponentially, then at some point, the amount of calcite that they have to add for a single chamber would become huge, and it might become really structurally difficult for them to do that. So maybe they just have to slow down the growth rates at some point. So that's something that we wanted to look into a little bit more. Oh yeah, so this is like a figure of how it changes through ontogeny. So it's fairly stable until about chamber 15. Ignore this bit. That was just when the first couple of chambers were forming and they hadn't had a complete whirl yet. So it stays pretty stable, and then at some point it starts to go up. So that means that the growth rate must be going down. So this is very interesting. The problem with this for our analyses is that it's a discrete measure. So it's just count numbers. And for statistics, it can be a little bit tricky. So we wanted to see if there was a continuous variable that we could use instead. So to do that, we looked at the angles between chambers. So when the number of chambers per whirl increases, it means that the angle between them increases as well. So here, for example, when there are 7 chambers in the final whirl, you get pretty big angles between them. Earlier on, when there's only 5 chambers, it's nearly straight angles. So that is a slightly more continuous variable to measure how growth rate, or sort of how their growth spiral is changing. So just comparing those two, this is the chamber number, and then this is the angles. And you can see that the angles actually start to increase a little bit before the number of chambers per whirl starts to increase. So it probably just means that when they grow, the angles gradually start to increase, which means that at some point there'll be an extra chamber in the whirl. But that shows that the angles are probably a more like biologically meaningful thing to look at. So we use that in our analyses for an indicator for shape. Yeah, and then comparing that to the cumulative volume, so just looking at the growth rates, I mentioned that they might be slowing down, the angles might be increasing because they have to slow down their growth. So I just wanted to quickly show the comparison, and you see that where the growth is slowing down here is pretty much the same where these angles start to increase. So they might be linked. That can mean two things. It could mean that they are both plastic traits and respond to the same environmental variables, or it could mean that growth rates are constrained by these internal architectural things. So to figure out the difference between the two, we reconstructed the environment at the time that every chamber was built, and then to use that to compare to both the growth rates and the angles between the chambers to see if there was a link. And we used laser ablation for that to target individual chambers. So here you can see these dots are the shots made by the laser. Again, Alex, our research technician, did most of the work for this. And then we looked at magnesium-calcium ratios because they're an indication of the sea surface temperature that these things grew in. These are just some examples from modern forams. So you see that magnesium-calcium in the shell increases with increasing temperature. The exact relation varies between species. So we don't know exactly which specific magnesium values in our forams corresponded to which temperatures because these things are extinct. We can't do culture experiments with them anymore. But all we know is that higher magnesium means higher temperatures, and that's really all we needed to know for the purposes of our study. So we wanted to compare the morphology traits with the environmental data. And to do that, we use generalized additive mixed models. These are basically models that combine a couple of different linear regressions at different points in the data set. So usually with a normal linear regression, you just get one line through the data. But forams go through a couple of different life stages, and they might not be linear, or at least they might not be the same. So you can see here in this data set, just the angles going through the ontogeny. Here they're pretty constant, and then they sort of shoot up towards the end of the life. If you plot a normal linear regression through that, it's not really going to do anything. But these generalized additive models are a lot better at just describing the data. So we wanted to know whether these growth rates that we identified with integral projection models were really important for population dynamics. We wanted to know if they were completely constrained by internal processes, just like architectural constraints in the shell, or if there was a response to the environment. So if it was, in fact, a plastic trait and it had a little bit more possibilities to vary that way. So we studied the relationship with the ontogenetic growth rates, ontogenetic stages, magnesium, calcium, and we also looked at interactions. So it could be that it's a combination of both plastic processes and internal constraints. And then finally, and this is probably the most important one, we wanted to know if there were differences between the species. Because if there were, that means that somewhere in the speciation process, something must have changed in like the plastic response of the species to the environment to create new species. So we used generalized linear mixed models for this. Generalized additive mixed models, the ones I mentioned before. We ran a couple of different models with different traits in it, and then we just wanted to see which model was the best at explaining the data. And to pick the best model, we used the Akaike information criterion, which is basically describing a trade-off between how complicated the model is and how well the model fits. So ideally, you want a model that fits perfectly, but if it has to be incredibly complicated, it doesn't really mean anything, and you can't make any sort of generalized conclusions. So these AIC values sort of introduce a penalty for how complex the model is. And what you want is this value to be as low as possible, because that shows that there is very little penalty. You have a good fit and relatively uncomplicated. So we just compared loads of models and looked for the lowest AIC score. Starting with the growth rates, the first model we looked at was just looking at growth rates versus ontogeny. So chamber number, it could be that. at specific chamber numbers, they start to change, and that would mean it is an internal constraint. Second model we tried was the same, but with differences among species. And then we did one that was both ontogeny and the environment. So we got magnesium, calcium in there. And again, without species-specific effects and with species-specific effects. And in both of these cases, by far the lowest AIC values are the ones with the species-specific effect. So that's very interesting. There's something in the speciation process happened that made them start to respond differently. And then the model with the environmental data with the magnesium calcium performs marginally better than the one without. You can see this is like the very definition of a convenient p-value. So there is definitely a little bit of an effect of the environment. So growth rates are both constrained by internal genetic influences, but also by the environment. And then finally, we wanted to do the same thing for the growth trajectories, for the angles between the chambers, same approach. But in this case, we also included size of the shell at different growth stages, just to see if there was a size threshold for changes in the chamber angle. So maybe at some point they get too big and they have to start reducing their growth. So we wanted to see if that was the case. And then again, also seeing if there was an environmental effect. So these are slightly couple more different models. So we've got size in red, just ontogeny in blue, environment, magnesium, calcium in yellow, and then both magnesium, calcium, and size in gray. And again, with and without species-specific effects. And in this case, the models with the environment in it, so the yellow ones and the gray ones, they don't really do very well. It's actually the ones with just the size-specific effects, including species-specific effects, that does the best. So there is a size-specific threshold for the growth trajectory to start changing and to go from isometric growth to something that's different. And it varies among species. So somewhere in the speciation process, things did get, the constraints that were there did get overcome. So to summarize this, this study over the last couple of years kind of made a framework of how we can study developmental plasticity in the fossil record by looking at both morphological changes and environmental changes. And we showed that at least for growth rates, they are influenced by the environment. It is a plastic trait, and that somehow that must have given rise to these 3 different species. But that doesn't go for shell shape. That is just completely driven by internal constraints. Yes, that is one way to tell these different processes apart. And then the next step is that we want to sample through these speciation processes and see where these differences exactly arise in the speciation process, if it's before speciation or something during or afterwards that sort of refines the differences between the species. So yeah, there's still quite a lot of work to do. So that was my project from Southampton. And then for the last couple of slides, I just wanted to briefly touch upon what I'm going to be doing at Yale. Slightly different. I'm going to be looking at the old CliMap collection. So you might have heard of CliMap. When I started, I had heard of it somewhere, but I didn't really know what it was. So it's basically the first estimate of past sea surface temperatures during the last glacial maximum. So in the 50s and the 60s, all these old research ships went all over the world, collected loads of sediment cores from all different ocean basins. It's really good spatial coverage. They collected loads of foram assemblages. They matched them to the temperatures that were like prevalent at those sites at the time. Then they collected last glacial maximum samples, did the same assemblage studies, and used those assemblages to sort of figure out how cold or how warm the water must have been in the last glacial maximum. And they found that sea surface temperature were roughly about 3 degrees cooler on average. And that was the first actual estimate of past sea surface temperatures. So this was published in the 70s. It's got loads of these beautiful assemblage slides. But then after that, the collection kind of got forgotten about because all the data was published. Nobody was really working on the slides anymore. As time went on, geochemical processes emerged that could be a little bit more like accurate at estimating sea surface temperatures in different places. So this collection kind of got forgotten about. It ended up at Brown a couple of years ago. It ended up in a storage cupboard. The storage cupboard was being cleaned out and the slides were going to be thrown away. But then they got rescued by the Peabody Museum at Yale, and now we can do all sorts of new stuff with them. Because now we have all these really cool imaging techniques that can do really high resolution reconstructions of like size and shape and just morphology of all of these specimens. So what we've got on top of all the assemblage data all around the world, we also got data on morphology, so size, shape, lots of stuff. So what we can do is like have a really high resolution track of how species change within their biogeographic range. So if specimens at the core look very different from those at the edge, how this changes. So it's a really great resource. And at the moment, I've got an army of undergraduate students just imaging all of these slides and doing all the size and shape analyses. This is the spatial cover that we've got, at least for the modern. As you can see, it's quite good. I mean, as usual, the North Atlantic is probably overrepresented, but there's really good coverage in the Indian Ocean as well, and in the South Atlantic. And even in the Western Pacific Ocean, you've got a pretty decent latitudinal transect that we can probably use for loads of species. So that'll be a really cool thing to do. And then we're also going to compare that to, for example, the PRISM slides from the Pliocene. And we're going to be comparing this to the last glacial maximum as well. So just to see if things like geographic range size, abundance throughout the range, size, shape, all of that together, and then see if we can just make maps for all the different species and try and predict, especially going back through time, if we can make like sort of a multivariate framework for things like extinction risk or expansion, or which species are going to be most successful. If anyone here is interested in that, the Peabody Museum does have a couple of small grants available for students to come over and do like a research project with these things. So if you're interested in that sort of thing, either contact me or contact Shelley Hull, who's working on that stuff. And yeah, that was it. So happy to take any questions.
Hi, that was really cool. I come from the climate science world, and so I was wondering if the last two slides you were talking about, if your work on looking at these species could that also inform like the sea surface temperatures? Like, do you see what you're finding from this project maybe like make some of the sea surface temperature data more accurate? Like, do you see it working that way too?
There's definitely a possibility for that. So we've got all the transfer function data for all of these sites with the different assemblages and the different sea surface temperatures. If we did, for example, magnesium calcium on the same specimens, we could see how far off these two different methods are, and if there's any chance for like refining the methods. It's not something I'm planning to do anytime soon. So if you're interested.
Well, thank you. I don't think I can show it.
Thank you. Good throw. So it's really cool to think about how internal processes might kind of control or at least like constrain evolution.
Yeah.
And these are, I'm trying to remember when these Menardellids actually evolved. But are these transitions related to climate transitions, or is this kind of like background?
I suspect it's background because let's go back. So they originated, or these couple of species originated in the middle Pliocene.
Right.
Oops, that one. fairly warm around that time,
Mhm.
but it's all tropical species. Their environment is fairly stable. I don't remember there's anything like a specific environmental things that might have caused the speciation. Also, if you do morphometric studies among these species, there's actually quite a lot of overlap between them. So it can be really hard to tell how far they're actually separated into species. But yeah, the outliers are definitely, or like the end members are definitely different.
Yeah. So I know that you said that kind of a next step of this project would be to look at like transitions and actually like the speciation events. Would you predict that there would be different drivers of shell morphology with actual like external climate driven extinction or speciation events?
I definitely think it's an interplay of different factors.
Yeah.
If the climatic if the climatic perturbations are severe enough, then I think most constraints can be overcome, except for like the most basic things that make a foram.
Right.
So yeah, I definitely think there's a combination of factors at play. And if there's not a lot of climate, if there's not a lot of environmental variability, then there's not really any reason for constraints to be overcome. If it works, why change it?
Right, exactly. And that's where the background, right, really comes in. Yeah. But yeah, it's really cool to think about the actual mechanisms that cause speciation and
definitely.
how they might vary. Yeah.
And usually for forams, it's quite hard because there's a lot of speciation happening, and most of the time it's not. It's not a huge amount of weird stuff happening.
The popcorn's a different shape.
Yeah, exactly.
Thank you. But following up on both of those, I was wondering how long is each one of these individuals like living and recording its environment? I was thinking just about when you're measuring the magnesium calcium like per chamber, if that growth is happening like relatively fast, I wonder if you don't see that much variation in the magnesium calcium because the temperatures are kind of like stable. But then that made me think of the time you might see it would be species that are sort of like living through a seasonal transition or living through some kinds of variability that aren't really like climate transition time scale, but might be much shorter time scale. So I was wondering if like from all of those different specimens, do you see different populations that maybe lived at like different times of year or lived across like different kind of seasonal temperature fluctuations?
Yeah, there's definitely a possibility. The nice things about these guys is that they're all tropical species, so seasonal variation isn't huge. And they build a chamber every 2 or 3 days or so there might be a little bit of day-to-day variation. It's definitely possible because they live for a couple of months that they go through some sort of changes. But especially, or at least in the modern, they kind of live year round. So some specimens live slightly warmer, some live in slightly colder conditions. It just really depends on very local, temporal changes.
Okay, can I ask a follow-up question? Just what do you think? I'm thinking about there's just the different kinds of timescales that are involved in like a foram's life versus like so much of what they've been used for historically. Like you mentioned in this last part of the project is like reconstructing climate 25,000 years ago or 3 million years ago and thinking about climate transitions that are not occurring over a 3-month foram life timescale.
Yeah.
So I wonder just, but it's also so powerful that they live for such a short time because there's so few kinds of proxies that give us that, that we know are telling us about a short window. And so I just wonder, do you think that from these kinds of studies, you can learn something about the past seasonality? And do you think that like, what is actually a more important, sorry, I'm formulating this question as I speak. And Adam, we'll get to you next. Do you think that these like large scale climate transitions, the Pliocene, last glacial maximum, or actually like changes in seasonal variation, is there a way to tell which of those is a stronger driver for the sort of speciation that you're looking at?
Yeah, that's a good question. This study just shows that these things just respond to like things throughout their life, so a couple of months. And then you can use that to sort of predict what will happen through a longer climate transition. So if we know that they respond to environmental changes, then you can use that and you see a different, or you see speciation through a climate change interval or something, then you can use that to say that, okay, it was quite likely that this thing did respond to the environment throughout this transition, and then that sort of gave rise to the new species. Yeah, hope that answers the question.
Thank you.
Yeah, so I'm going to preface this by saying I know nothing about forams. So I don't know if maybe some forams like don't grow as long as others and will have different numbers of chambers despite being the same species. So can your method sort of also inform about like identifying different species, or is it sort of that like less of an important thing?
It definitely is. There is a little bit of variation in how long species live. It's in the order of magnitude of a couple of chambers. So they've got something between 17 or 21 chambers, for example. That number is not usually used to identify different species. Usually when people identify these by looking down a microscope, so you can only see sort of like the external final whorl. Sometimes you can see a little bit of the previous chambers, but usually not enough. So species differentiations are usually made just by what we can see on the outside. But it's actually a very interesting question, and I've got a grant proposal in looking into like more these sort of life history differences between species. So yeah, definitely a lot, hopefully.
Really question, so you show a change in the angle of the chambers at one point, and I don't know if I followed very well, but it might be due to some environmental factors or other factors, but could it just be a geometrical problem in the fact that the shapes demand for growth to change angle? It's just internal problem, not at all external.
Yeah,
Is
yeah.
it possible?
Yeah, definitely. And especially the results that we got from the generalized mixed models, there was no effect of the environment on like the changes in angles. So it is probably just an internal thing of how they built the shells.
So is it possible to model that growth and change in angle? That'd be really cool, right?
Yeah, I would absolutely love to do that, and I try to do that. There's things from, for example, ammonites and gastropods. I think it's called Raupian parameters, where you look at the inflation rate along the z-axis and along like xy plane.
Yeah.
I try to do that with forams, but it doesn't work very well because whoops, that way I'm going. So the things that Raupian parameters were designed for was something that grows continuously. So just a lovely, nice growth spiral going all the way around. When you look at this looks like a pretty nice growth spiral as well, until you start plotting actual logarithmic spirals next to it. Let's quickly go through backup slide. Yeah, so these are the Raupian parameters. This is a logarithmic spiral. This was the foram. So they grow in discrete stages. They're pretty wonky. And also they go through different life stages, which means that they don't have a single vertical coiling axis. I've tried to mathematically define the coiling axis of these things, but it wasn't possible. It was just, there's too many variables. There's too much variation.
Can I say something? It sounds like an optimal transport problem. Like you define initial shape and you end with a final shape, and you're trying to find what is the optimal map between the work with, like, to go back to mathematics basically and do that. Like, sounds like a really nice problem for that.
Yeah, that sounds really cool. I'd love to do something like that, and also just to get like a theoretical morphospace of what these things could look like and what they actually look like, and then compare the possibilities.
Thanks. Thank you.
Thanks. I think Adam has a question online.
Hello. Hi, everyone. Hi, Anieke. Great talk. As always. My question is regarding on your potential, what you're working on now and potentially working on in the future, and whether or not you thought about potentially looking at, because the climate, I guess, is pre-industrial kind of Holocene, and whether or not you thought about looking at plankton toe samples and comparing that pre-industrial to post-industrial Holocene and doing these same kind of analyses you're working on right now, looking at maybe how those changes have occurred over the last, well, since the Industrial Revolution.
I had not thought about that, and that is a brilliant idea. So yeah, that would be a really cool thing to look at, see how things have changed since the like Industrial Revolution. Maybe see if we can use our predictions going from the Pliocene to the recent, and then see if we can predict how the modern has changed. That would be really cool. Thank you for that suggestion.
Cool. Exciting. Thank you.
Does this work? Okay, cool. I was wondering for your kind of what was the like number of data points you had for chamber size and angle, and whether it was enough to get distributions at each of the different points? Because it'd be kind of, I think it'd be pretty informative to see what the distribution of different chamber numbers are, and like their sizing and angles, and see if there's, you know, are they perfectly symmetric? Are they leaning one way or another way? Are they maybe bimodal? Did you have enough data to potentially look at those distributions?
We do have enough data. One thing we found, especially with the angles, so these data is not actually the chamber number that seems to be driving things. It's the total size of the shell.
Okay.
So we could do something for different size classes and see like what they are like. Yeah, definitely.
We'll talk. Thanks very much. I'm so going to be the jerk that says more of a comment than a question. I'd love to introduce you to my friend Kathleen Ritterbusch at Utah, who is doing similar things with ammonites. And especially if you're thinking about putting them in sort of like an ecological, what space is possible, what space leads to more drag, that kind of thing. She's thought about all of this stuff with Westermann morphospace and ammonites. And she's doing some very similar stuff to you. I think you guys would have a lovely chat.
That would be awesome. Yeah.
Also, are these, I can't remember which genera like flip their spiral left and right. Do you see any difference between left spiral and
No,
right?
the very vast majority is all the same coiling direction.
Thanks.
Thank you.
Other questions? Questions online? Going once? Going twice? All right, well, let's thank Anieke again for a great talk.